Good evening, everyone, and welcome to Pitch Club. And thank you for the humor in the chat. I like that. We at AngelMD love providing our membership access to really compelling startups. And tonight we have a focus on therapeutics. If you haven't already, please introduce yourself in the chat and take a second to add your name, your location, and what you do professionally. As we've noted in our vital signs briefing, Pitch Club is going to be taking a break from June through August, but we will be returning in September with some terrific programs and format updates. We don't want you to miss out. In order to ensure that you don't miss out, we encourage you to register for the Pitch Club series. And when you sign up for that, we automatically register you for the events. It doesn't require that you attend, but it ensures that you are registered and you'll get all the communications around those pitch clubs, including the follow-up information and the recordings if you do end up missing the event. So we will provide the link in chat, but we will also provide it in the follow-up email that we send out tomorrow. So with that, I would like to introduce our sponsor. It's AngelMD Academy. I'm really excited to share with you about AngelMD Academy because it is our own training organization. The first course launches this week. We've conducted countless educational programs over the last several years, but now we have our own curriculum and educational content from partners, and it'll be available 24/7 for members. If you can attend live training programs and access on-demand content, programming will cover topics for entrepreneurs, startups, board members, clinicians, investors. We're really trying to make sure that all of you get some great education programs. If you're a lifelong learner, we encourage you to participate. And so, as I mentioned, our first live training will be this week. And so I'd like to introduce Dr. Saya Nagori, and she's going to be telling you about the first course for which she will be the instructor.
Hi, everyone. Thank you, Jennifer, for that introduction. It's so great to be here. I am so excited to be part of AngelMD and AngelMD Academy. I just want to give a shout out to Tobin for bringing me on. We had a wonderful conversation last year, and we just had so many similar alignments in terms of how we can really bring unique and useful and thoughtful education to physicians, nurses, pharmacists, and other healthcare providers, because we see so many companies in the healthcare space. And one of the things that is not great sometimes is that a lot of these companies are not led by the clinicians that should be leading them. And so I have been a physician entrepreneur for the last 7 years. I started a company called simplehealth.com. It used to be called simplecontacts.com. I am an ophthalmologist, and I developed the first remote eye exam that was used in the United States. We do have a patent on that. And Simple Contacts grew to about 50 people. We were operational in 42 states until we did a hard pivot into women's health. And that actually made the company scale even faster. We ended up dropping eye care. And now simplehealth.com is almost 200 people. And we are growing fast. And I've moved from chief medical officer to an advisor. I was VP of medical affairs. And that was really just the beginning. I also advise for a company called Wheel.com, which is a B2B player in the telemedicine space. I also advise for a company called HiDrB.com, which I'm sure most of you have heard of ZocDoc. So the founder of ZocDoc, Cyrus Masumi, reached out to me a few months ago, maybe about 6-8 months ago, wanted someone to come on to lead his ophthalmology initiatives. And I've just been really lucky in some ways, but I created a lot of that luck. And that's what I really want to share with you guys, is how to be ready for when those opportunities present themselves, so you can be the one that they choose for their company. So I'm really excited to be leading this. I get so many messages about how can I get involved in industry. And it's not something that happens overnight, but certainly it is something that everyone is capable of doing. And this world, this startup world, the world that AngelMD encompasses, it's changed my life in the last 7 years in a fantastic way. So I could literally talk about this for hours, and I know you guys have to get to Pitch Club. So I'm happy to answer questions via instant message. You can DM me on Instagram if you have any questions about what I'm going to cover in the course. And certainly you can ask the AngelMD team as well. So I will say goodbye for now. Hope to see you guys on Thursday.
Thank you so much, Saya. And really excited to have you as a part of AngelMD and AngelMD Academy. I put in the chat, angelmdacademy.com is where you can register for this upcoming course. As Saya mentioned, the first one is this Thursday. So we encourage you to register as well as send it to your friends, because as Saya said, much of being an advisor for startups is very purposeful. And I think she's going to give us some secrets on how she got to where she is. So I'm super excited about that. Thank you so much for being here.
Thank you.
We're going to move on to our presenters of the evening. So as Jennifer mentioned, tonight is our last Pitch Club until the fall. And we have 3 amazing companies in the field of therapeutics. Our first company is called SFA Therapeutics, and we have Dr. Ira Spector, who is the CEO of SFA Therapeutics, here to tell us about their company and answer some questions for us.
Spector, the CEO and co-founder of SFA Therapeutics, where we have a very big vision to change medicine by treating the root cause of autoimmune disease instead of the current gold standard of immunosuppression. And we're developing this with psoriasis as our first indication, but our plan is to go after numerous autoimmune diseases. The current therapies for psoriasis, as you probably know, are insufficient. The responses aren't durable, efficacy is limited, or if it's good, it comes with a cost of very high adverse events. Most drugs are antibodies, which cause antibody formation, which leads to limits in treatment. It requires injections and infusions that are painful and inconvenient and leads to a poor quality of life and a high cost per patient. In fact, most patients are unhappy with current therapy, and only 6% of patients take the drugs you see on television every night, because for the other 75 to 80%, the treatment is worse than the disease. We plan to expand the market to that 80% with a new drug that is both efficacious, highly efficacious, and highly tolerable and highly safe. So our better solution shows a durable effect. In fact, 3 out of the 6 patients in our phase 1A translational trial are off of drug with no recurrence of symptoms. We're showing excellent clinical efficacy. In fact, we have patients such as the one shown here who was treated orally for 45 days and is shown after at 4 months, who are off of treatment with no side effects, with an oral drug that is potentially disease modifying. And in the Q&A, I'm happy to explain why we think it's treating the root cause. And we think this is going to lead to major quality of life improvements. And of course, we are aiming at additional indications like RA, psoriatic arthritis, ankylosing spondylitis, ulcerative colitis, Crohn's, etc. Here's another subject. This subject was treated for a year and has been off treatment for over 7 months with a greater than 90% clearance and no side effects. So how does this work? It works through 2 primary mechanisms. The first is signaling through G protein coupled receptors to specific receptor sites throughout the body. And the second is in the nucleus through transporters. Now those receptors are immunomodulatory, meaning they inhibit pro-inflammatory IL-17, IL-12, and IL-23, plus they upregulate anti-inflammatory IL-10. And in doing so, they also inhibit interferon gamma activity, and I'm going to get back to that in a second. And in the nucleus, this is a potent HDAC1 and HDAC2 blocker. Now that's important because it degrades the mRNA of TNF-alpha, which is a major signaling component for inflammation. And that's why this is so exciting. In fact, if you look at the etiology of psoriasis in particular, regardless of the trigger, it's caused by stressed keratinocytes that upregulate IL-12, IL-23, TNF-alpha, and IL-17, manifested as inflammation. And the lack of regulation of interferon gamma leads to plaque formation of the keratinocytes. We downregulate all of these channels, which makes this the ideal fit for psoriasis, but also these cytokines are implicated in other diseases, drugs. Now, in our first trial, 6 out of 6 subjects showed significant lesion clearance, and they all had different demographics, different locations, different backgrounds, but they all showed significant psoriasis area of severity index clearance in a fairly short period of time. And that's why we think this is a complete game changer. So a little bit about the company. We were incorporated in 2016 based on an initial discovery at Temple University in 2015. We received our first patents in 2018 and 2019. Our investigational new drug authorization was received in 2020. And by the way, in the pre-IND meeting, the FDA called this novel. We received our seminal patent, which is really foundational to the company, in 2021. We're currently in a phase 1b clinical trial in patients in 30 subjects, 15 in our first formulation, 15 in a newer formulation. And the better of those two formulations will go into a phase 2a clinical trial with 60 subjects that the FDA has already approved. We think that's going to lead to our first potential exit somewhere toward the end of 2024. And then we plan to develop additional indications in diseases like rheumatoid arthritis, psoriatic arthritis, ankylosing spondylitis, etc. Our key investors include North-South Ventures and AngelMD Capital, among others. And we think that we are a potential company to either license or be acquired by the major pharmas shown here. We're a proven management team of drug developers and commercialization experts. I'm the S in SFA. I've worked on 34 approved drugs. Mark Feidelson is a world-class immunovirologist who's the F in SFA, and Ala Arzumanyan is a world-class microbiologist. King Lee is a head of regulatory, is a pharmacologist. Jim Kerwin is our head of clinical operations, was my right hand at Wyeth for 5 years. Sean O'Brien is an industry veteran who's helped sell 6 biotechs, and Rob Dickey, who's ex-Lehman Brothers, is an industry veteran. We want to change the way the 145 million patients with psoriasis are treated, and then change the way the 350 million patients with autoimmune disease are treated. Please join us on this very important journey. Thank you.
Wonderful. Ira, thank you so much for telling us all about SFA Therapeutics. And thank you for being here tonight.
Thank you.
This is very cool. One of the things that I like about Therapeutics Night specifically is we really dig into the science. So I appreciate it. It takes me a little bit back to medical school, which might have been a couple years ago, but it's still fun to remember some of those things. So very informative. Speaking of that, talk a little bit more about how your pathway differentiates this particular drug from the current drugs that are on the market.
Okay, so I helped develop one of the drugs that's on the market. I ran the clinical trials for Enbrel. I was there when we had the first cases of TB and the first cases of cancer, and we had to figure out were they real, and they were. And those drugs work through immunosuppression. They just knock the immune system completely down, typically with one channel. So Enbrel is TNF-alpha, and some of the other drugs on the market are other channels. Big Pharma works on a single channel, single target mantra. This is a very interesting drug. This drug was found in the GI microbiome, originally made by bacteria, isolated, synthesized organically, and turned into an oral drug. What we found, and half my training is epidemiology, is that patients who have autoimmune diseases rarely have one. They almost always have 2 or 3. And oh, by the way, clinically, if you take a good history, they often have GI issues, IBS, IBD, Crohn's. We believe that these substances, originally made in the GI microbiome, should be passing into the bloodstream, and that their role in the human body and physiology is to immunoregulate. They have very short half-lives, very short PKs. We figured out how to create sustained blood levels. And we believe that if you give these substances, that you provide exogenously what the body can't provide in these patients. And just like you give insulin to type 1 diabetics because they can't make it themselves, we think you can give these to patients with these clusters of autoimmune diseases. And that's why we're seeing some startling results.
Very cool. Thank you again for explaining that for us. We have one question in the chat. Again, you can put your questions for Ira and SFA Therapeutics in the Q&A. And Q&A is just to the right of the chat. It makes it easier for us to filter through those questions. So if you'll put them in the Q&A, I would really appreciate it. But one of the questions really is around side effects that you may have seen, and if there's any neurotoxicity at all.
We've seen no neurotoxicity. We do believe that at high doses, based on the formulation, that we may expect to see some constipation, but we haven't seen it in the current trial or the previous trial. We have done extensive reviews of the toxicology. Obviously, we can't be in humans without having provided a safety package to the FDA, which was done in 2018. And we have seen no neurotoxicity associated with this.
Fantastic. And you mentioned in your talk that coming soon, you're going to look at other indications for this drug. Tell me more about that.
Okay, if you look at the cytokines that we're downregulating, TNF-alpha, IL-17, IL-23, interferon gamma, and you do a systems biology review and you map those 4 cytokines across other diseases, we stopped counting after 20 diseases. We think that there's a cluster of autoimmune diseases that are all related to the same defect. So after psoriasis, we're going to go after RA, because if you have, so I'm an epidemiologist and biostatistician. If you have psoriasis as a child, you have a 50% chance of RA as an adult. If you have psoriasis as an adult, you have a very high likelihood of psoriatic arthritis later as an adult. These diseases are interrelated to the same inability to regulate these pro-inflammatory cytokines. So we believe there are a number of diseases that will be treatable. And current medical care is fragmented. So currently, dermatologists will use the latest thing that comes out. So they tend to have all gone to IL-23 agents, whereas rheumatologists tend to use IL-17 agents like Enbrel. However, we believe they're fundamentally the same disease, and the human body has parallelism. So if you downregulate one channel, you may find there's crosstalk and feedback, and another channel goes up eventually, and the drug stops working. By downregulating all 4 channels simultaneously with the same substance that should be there if you're healthy, we think we've found a way to match these against these diseases. So that's why we're so excited.
Well, yeah, what you're telling me is this could be a game changer, which is really exciting. Thank you so much for being here. Thank you for telling us about SFA Therapeutics. I know Ira's going to stick around for our networking after the event, and he will be there to answer any additional questions that you have, but really exciting. I want to make one quick reminder. So after each presenter, we want you to fill out a poll. This feedback goes directly to the companies and the CEOs and founders. We leverage Poll Everywhere, and I'll put a link to that in, well, there we go. We have a link to that in the chat. So if you go ahead and click on that, we're going to be using that poll after each presentation tonight. Please, 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 even if you're not a physician, fill out the poll, because these companies want your feedback. But we're now going to move on to our speaker number 2. So we're going to hear from Anil Asrani, and he is the president and CEO of a company called Tier Solutions.
My name is Anil Asrani, and I'm the CEO of Tier Solutions. A big thank you to the AngelMD team, and of course, to all of you for the opportunity to be here with you today. Tier Solutions is a clinical stage biotech. We have impressive human clinical data, robust IP, and a clearly defined clinical and regulatory path with formal written FDA alignment that supports an unrestricted general dry eye indication, and that's for our lead product called Lacropep. For those less familiar with the dry eye market, this is the same path that a dry eye drug called Xiidra went down, and they were sold to Novartis for $5.3 billion. Our first in human data has us and others convinced that we are better than Xiidra on multiple fronts. And the reasons that you see these kinds of valuations in dry eye is because dry eye is essentially the hypertension of ophthalmology, cited as one of the top reasons someone would go to an eye care professional. But unfortunately for patients suffering from dry eye, which is a large and growing market, the current products do not provide great solutions, which fortunately for us leaves this market susceptible to disruption because the problems are well known. For example, the major products have been designed to work by suppressing the immune response in the eye, which does reduce inflammation and makes some people feel better, but it does not address the underlying cause of the disease. Rather, it puts a Band-Aid on the symptoms. And these products take too long to provide relief. And when used, patients are often complaining of a variety of negative side effects. And to make matters worse, after using these products for months and enduring this discomfort, they fail to work in a majority of patients. Lacrotin, on the other hand, is our proprietary protein, which was discovered by using a screen of the Tier proteome. And the goal of that screen wasn't to find another immunosuppressive. It was to determine what was the actual biological cause of dry eye. In other words, what was missing in dry eye tears as compared to healthy normal tears? And the answer is lacrotin. It's deficient in all forms of dry eye. And that's not just us saying that. It has replacement therapy potential, which is what gave rise to Lacropep, which is pictured in the top right corner here. Lacropep is a peptide that represents the active binding domain of lacrotin, thereby preserving all of its cellular signaling bioactivity. And that has been extensively studied and published in peer-reviewed journals. Lacropep rapidly repairs and regenerates the ocular surface, even in the presence of inflammation. And only 3% of patients studied reported any discomfort when using our drop. And our therapy works in a majority of patients. Now, nothing, whether approved or in the pipeline, comes close to this kind of overall performance. And the reason for our success is due to how we solve for the underlying pathophysiology of dry eye. To get right to it, the cells of your cornea have no blood flow. They are avascular, no capillaries. So how do these cells even survive? Well, the tear film essentially serves as their blood. It provides everything those corneal cells need. And the major components of the lacrimal functional unit, which we're showing here, are specifically designed to keep the tear film optimal, thus keeping those corneal cells alive. But when any one of these components fails for any reason, dry eye occurs. And this is particularly true as it relates to the corneal sensory neurons, which we'll pick up here in a second. So let's use an analogy here to make this simpler. If you were to think about the components of the lacrimal functional unit as an orchestra, and each of those components as a section in that orchestra, then lacrotin is the conductor of that orchestra. As you can see, lacrotin is produced by all the cells and glands highlighted in green, and it binds its receptor, syndican-1, which we know is found on lacrimal acinar cells in the lacrimal gland, corneal epithelial cells, and corneal sensory neurons. And through that interaction with syndican-1 and a G protein coupled receptor, co-receptor that we identified, that's how lacrotin and Lacropep open the door to a whole new class of drugs that actually goes well beyond dry eye indications. But in dry eye, we have extensive peer-reviewed publications showing that Lacropep's mechanism of action optimizes all three layers of the tear film. Not only that, in the presence of inflammation, it protects the corneal cells from the damage that inflammation can cause, and its cellular signaling helps repair and regrow not only corneal epithelium, but corneal sensory neurons as well. And as I mentioned on the last slide, these neurons are critical because they provide the biofeedback to the components of the lacrimal functional unit, in order to ensure that they keep that tear film optimal, thus keeping the cells of the conjunctiva and the cornea alive. So it was for these reasons that we were so bullish on Lacropep's disruptive potential, and we're excited to get it into preclinical. But now let's move from the theoretical to the practical. Here you get to see the speed and the power and the promise of Lacropep in a photo that comes from our preclinical studies, as well as from data from our first in human clinical trial. Now, the preclinical is an autoimmune mouse model that represents a Sjogren's syndrome dry eye model, and you can see a heavily diseased corneal surface as evidenced by the staining here on the left. And after just 3 weeks of treatment with Lacropep, you can see a completely healthy corneal surface on the right. Moving to our first in human data, which was in a primary Sjogren's patient population, who are often excluded from dry eye trials because they have a very severe form of dry eye and is very difficult to treat. We showed a statistically significant improvement in an FDA approvable sign, which is an objective measure of ocular health at both 2 and 4 weeks in our low dose. And we showed a statistically significant improvement in an FDA approvable symptom, which is a subjective patient reported measure at 2 weeks with good trending at 4. And through our company's history, we've continued to make great progress. For example, we've been and continue to be supported by significant non-dilutive NIH funding. And early on, we attracted investment from Santen, a leading Japanese ophthalmic strategic. We have ongoing collaborations at leading institutions like Harvard and Berkeley, amongst others. We won the first tier Film Dues 2 Innovation Award, and we've recently been chosen to present at highly selective ophthalmic conferences. As for next steps, with our first in human behind us, we're currently raising $25 million to get Lacropep back into the clinic to conduct the first of 2 phase 3 trials for an NDA submission. And while we're already in confidential discussions with major multinational strategics, we expect an exit to one of the majors within 1 to 2 years. Representative examples are included in the top right. And we have the right team with the right set of management experiences to get us to this exit. For example, we have the former head of R&D at J&J Vision's Consumer Eye Health leading our development, as well as some of the same people who brought the most trusted laser and LASIK to market. But to summarize, the real story here is around our founder, Gordon Laurie. It's also around the lacretin protein that he discovered and named, and the deep scientific foundation and product development that is attracting the attention of major multinational strategics and clinical luminaries alike. If you hear nothing else from me today, please hear this. Our thesis is that lacretin is to dry eye disease as insulin is to type 1 diabetes. If it's not being sufficiently produced, then you have the disease and you will need lifelong replacement therapy. And for dry eye, we believe those are going to be coming from our company, Tear Solutions. Thank you for your time today. I look forward to Q&A.
Amazing. We
Thanks,
got
Katie.
a lot of wows. We got a lot of clapping hands. This is really exciting. And as several people have put in the chat, we have a stellar group of not only presenters, but I would say therapeutics tonight, with Lacropep being one of them. So tell me a little bit more about the efficacy of Lacropep, as well as, again, those side effects that the other drugs have that you're really trying to avoid.
Sure. Well, I think what's really strong for us is the speed, right? It's the speed with which we're providing relief to these patients. Typically, other therapies take months to provide relief. And as you mentioned, most of the major therapies that are out there come with side effects that are quite undesirable, meaning more than a quarter of patients are reporting that they're burning and stinging when they put the drop in their eye. Xiidra, which I mentioned, which was a big sale to Novartis, actually has another one against it. It has burning and stinging, but it also has dysgeusia, which is where you put the drop in your eye and you get a foul metallic taste in your mouth that lasts for hours. The other thing, there are steroids out there that are fast acting, right? But the problem with those steroids are you can take them for a couple of weeks, but then you have to stop because you'll get an increase in intraocular pressure. So you're essentially trading dry eye for glaucoma. Obviously not a great trade. So our speed, our safety, and most impressively, that we're getting a response in a majority of patients is really what sets us apart from the competition, where you see slow acting, high side effect rates, and the inability to treat a majority of patients.
Sure. No, I mean, again, I think this seems like another game changer. One question, because you brought up Xydra. We have a couple ophthalmologists in our out in our audience tonight. So Dr. Nagori said one of the problems or concerns with Xydra that she has with her patients is that insurance isn't covering it, and many patients can't pay out of pocket. Tell us a little bit about how that's going to be different with Tier Solutions.
Yeah, well, I think the way, so when you think about the clinical data coming from Xydra versus Restasis, there has been no study that I'm aware of where Xydra went head to head with Restasis. So they did not show superiority to Restasis, and that's really the key that unlocks better and more comprehensive coverage from a reimbursement perspective. So our plan right now, obviously, is to get Lacropep back into the clinic, look for our optimal dose, right? And then once we have optimal dose, go and take that optimal dose head to head against the entrenched competitors, which would be either a Restasis or Xydra, and then look for superiority. If you get that superiority claim, that again opens up a lot of doors from a reimbursement perspective. But the other thing that I should mention is our current plan is not to take this to commercialization, right? Our plan is obviously to be working with strategics who have that muscle from a reimbursement perspective and look for an attractive exit. If we can't find the attractive exit, then obviously we'll build the company out to commercialization. But more than likely, this is going to be taking a partnership in order to make this.
I have a feeling you're going to have some interest. I mean,
yeah.
I'm
the interest is already there. That's the nice thing. The interest is already there. And what we're trying to avoid, we've not had to give away any strategic rights in our company so far. And we would not prefer to do that. Obviously, when you start to talk to the strategics, that's what they're looking for, because they're looking to get an attractive deal.
Right.
And so from our perspective, what we'd like to do is run this a little bit further ourselves and really maximize the valuation in the company.
I love it. So we have time for a couple more questions from Dr. Jennings, another ophthalmologist in our audience tonight. Does Lacropep also help stabilize evaporative dry eye based on meibomian gland dysfunction?
Oh, that's a great, I'm so glad I couldn't have paid for a better question, actually. So we've actually got a peer-reviewed publication that we did with a world-renowned lipid expert out of Bulgaria. And essentially, what we were able to show is that Lacropep actually has a physicochemical property to it. So you can take normal tears that would not be susceptible to collapse, and you can selectively remove Lacropep. And there's a longer story. You're probably going to say Lacropep is in the tears. Yes, it is actually. I'll tell you a little bit about that in Q&A in the breakout. But we've taken normal healthy tears, selectively removed Lacropep, and they will collapse. We've then taken dry eye tears that are susceptible to collapse, just added back Lacropep, and they maintain the structure and do not collapse. So this is really, fascinating. It is. We know it's interacting with a particular molecule. We can get into the weeds on this in the lipid layer, and that's really what's allowing it to stabilize the lipid layer. So that stabilization of the lipid layer prevents the evaporation of the tear.
That's amazing. As I said, after SFA Therapeutics, one of the best things about Therapeutics Night is the science and the medicine runs deep. So very cool. I have one other question, just because this blew me away a little bit. I think you said in your presentation that Lacropep promotes neuronal regrowth. Is that true?
So so what I will tell you is we have the science to show this. Expect that to be coming out in peer-reviewed publications soon. But yes, we are finding that this has the ability to not only preserve and repair a barrier function in the eye, but also help regenerate essentially and regrow corneal sensory neurons. Now, the really exciting thing about this is LASIK procedures, as well as other refractive surgical procedures, obviously damage those nerves when you're doing those procedures. And we believe that we've got to play there in terms of that wound healing and getting those nerves to regenerate quicker, and obviously then help those patients after those surgical procedures, because typically, and again, the ophthalmologist would have the data closer to them than I would, but I think roughly speaking, 90% of people that come out of post-refractive surgery have dry eye for at least 2 or 3 months. And so we think we can speed that process up.
Yeah, no,
And maybe avoid it altogether, but we don't know that yet. We have to study.
Yeah. I'm going to come drop by your table in networking, because the other thing that I want to know, but we don't have enough time for it right now, is when you talk about that, I go to, couldn't this technology be used for other indications? So more to come on that. I'll let you answer that question in networking. Thank you so much for being here and telling us all about Tier Solutions. Again, we're going to put in the chat for you the Poll Everywhere link, because Anil and Tier Solutions would love your feedback. Yes, and amazing job. Thank you for being here. Super exciting for all the dry eye sufferers out there, which my understanding is there's a lot. So
thank you so much for having us, Katie. This was a genuine pleasure. Thanks very much for the time.
Thank you. All right, so we are going to get on to our last presenter of the evening. And we have with us Pedro Correa de Sampaio, and he is the co-founder and CEO of Neob Therapeutics. So he's going to tell us about another exciting therapeutic.
Hi, my name is Pedro Sampaio, and it is a pleasure to be here to tell you about the work that we're doing at Neob. Immunotherapies have completely changed the clinical landscape of cancer by harnessing the immune system to eliminate cancer cells in these patients. The problem is that we're still stuck at this number of 80 to 85% of patients with solid tumors that do not respond to this therapeutic strategy. A subset of these patients can generate local barriers to the infiltration of immune cells, which get trapped in interstitial regions and are unable to get close to cancer cells to be able to do their job. These are known as immune-excluded tumors and can make up to 50% of some solid tumor indications like microsatellite stable colorectal cancer, pancreatic cancer, triple-negative breast cancer, which are particular unmet clinical needs that don't respond to immunotherapies. Now, within the microenvironment of these tumors, we know that the local fibrotic extracellular matrix is a particularly important mediator of immune exclusion, creating physical barriers to the infiltration of immune cells into the tumor core. For this reason, there have been several attempts to try to address extracellular matrix within the tumor. Originally, some companies tried to develop drugs that are systemically delivered to these patients to remodel extracellular matrix components, which have two main problems. One, the drugs themselves struggle to get through to this highly fibrotic environment, but mostly these extracellular matrix components are common to healthy tissues. So systemic delivery of these drugs can lead to pretty severe toxicities, and the drugs have to be brought down so much that they end up not really doing much to the tumor. For this reason, new strategies have arisen, like the inhibition of pathways that can lead to the production of matrix like TGF-beta through antibodies or multikinase inhibitors. The problem with these strategies is that you're inhibiting the production of additional matrix, but you're not really addressing the extracellular matrix barriers that are already in place. There's a clear need for new strategies that eliminate the extracellular matrix barriers to immune infiltration existing in the tumor without leading to additional toxicities. And at Neob, we think that we found the solution. And that is to genetically engineer tumor colonizing strains of bacteria to disrupt the extracellular matrix locally in the tumor, and through that enable immunotherapies in current non-responders. We are genetically engineering strains that have an innate capability of colonizing solid tumors, and we are engineering them to be activated by the conditions that you find within the local tumor core to produce a payload that targets specific extracellular matrix components that we know are involved in immune exclusion. The bacteria are activated, these payloads are secreted, and they remove these extracellular matrix barriers to immune infiltration, enabling the immune cells to get into the tumor and these patients to respond to immunotherapeutic strategies. Now, all of this is based on several years of research on all of the different components. We know particularly that these bacteria have the capability of colonizing tumors, and this has been extensively demonstrated in mouse models of cancer, that once you systemically administer these safe strains of bacteria, they colonize the tumor within 24 to 72 hours and get very effectively cleared from all of the rest of the healthy tissues. We've already engineered a bacterial prototype that we can induce to produce one of these payloads, and we can see that this payload can be effectively released into the extracellular space and target the matrix component that we are going after, reducing the extracellular matrix integrity in vitro. And we know from the literature and from several studies that have been done in the past that by reducing the extracellular matrix integrity and removing the barriers to immune infiltration, you can very effectively increase immune infiltration into these tumors. And this is just one example in a mouse model of pancreatic cancer, where by disrupting extracellular matrix integrity, you see all of these brown immune cells getting into the tumor, which weren't able to do so in the control mice. Now, how exactly are we engineering these prototypes? We're doing so in a modular approach using individual genetic circuits that we're then putting together for the development of our bacterial product. Each of these circuits includes the biosensor or the tumor-inducible promoter to make sure that the bacteria is activated by the local levels of oxygen or pH that you find within the tumor, the payload itself, which are enzymes that target specific components of the extracellular matrix that are important to block immune cell infiltration, the secretion system to make sure that we target the matrix directly through release of the payload into the extracellular space, and the antibiotic-free plasmid stability system to make sure that these bacteria are amenable for clinical application into patients. All of these individual circuits were assembled using the basic DNA assembly platform, which was developed by Professor Jeff Baldwin, who is the co-director of the Center for Synthetic Biology at Imperial College London and one of the advisors of NEOB. And using this platform, we have already built an extensive library of genetic circuits for all of these individual modules that will then be harnessed for the establishment of the platform for the engineering of different bacterial products that target the extracellular matrix. Now, we started only last year in the beginning of 2021, but while we are quite early, I think we've accomplished quite a lot in our first year. We started with funds from Deep Science Ventures and Cancer Research UK that allowed us to build all of these genetic circuits toward the development of our initial prototype targeting colorectal cancer. Through the process, we won several different startup competitions, including a place in the very prestigious Accelerated Abraham program here in Cambridge in the UK. We're currently working on our first patent application with Potter Clarkson, who is an expert in this particular field. And we've just won a grant from Innovate UK from the British government to initiate the development of our second bacterial product targeting breast cancer patients in collaboration with the Barts Cancer Institute here in London. We're currently raising a seed round to start the efficacy studies using ex vivo patient material and then moving into in vivo animal studies, for which we're also writing another grant application for the ERC with our collaborators at the University of Cyprus. After we go through our in vivo studies, our plan is to raise a Series A round, which will allow us to get into IND enabling studies towards a first in human combination study in 2026, 2027, which we intend to do in co-development with pharma companies within the immuno-oncology space. And we've already started contact with them, and they've shown interest in the development of our product. We've got the right team in order to be able to achieve this that has over 25 years of joint experience in both cancer biology and bacterial engineering. We've got a world-class panel of advisors with both academic leaders and former C-level executives in bacterial engineering. So if you want to find out more about what we're doing, please feel free to reach out, and I'll be very happy to take your questions.
Fantastic. Another really exciting new therapeutic. I think we'll get Pedro up here to the stage. Again, if you have questions for Pedro and NEOB Therapeutics, please put them in the Q&A. I'm going to start with a couple of my own, though, because I'm guessing there's a couple people in our audience tonight that are thinking the same thing. So genetic engineering of bacteria. This sounds a little scary, right? people get worked up about these things. How are you going to make sure that this drug is something that's going to help these patients and not harm them?
But yeah, that's a very understandable question and a common reaction. I think the first thing to say there is that we're not the only people doing this. The idea of using bacteria as therapeutics for cancer have actually been around since the beginning of the 19th century, when William Coley initially found that some bacterial infections could lead to complete remission in patients with sarcoma. And there are a few other companies that are developing bacterial products to fight cancer, both genetically engineered and not. We have obviously taken this into account in the development of our strategy in 3 main points. One is through the selection of the actual strains of bacteria that we're using, which are all class 1 safe strains of probiotic bacteria that lack virulence factors and that particularly are unable to perform horizontal gene transfer, so that we make sure that they're not able to transfer their genetic material that we're engineering them with to the
Sure.
local microbiome of these patients. But we're also, and I sort of alluded that in some of the modules that I was talking about, we're including the plasmid stability system that ensures that these strains of bacteria are also sensitive to off-the-shelf antibiotics, so that we can make sure that we can eliminate them easily if that is the case. And the final sort of points that we have is the possibility of engineering kill switches in the bacteria if after we do our in vivo studies, we realize that there's some persistence there after they sort of secrete their cargo, and that we need to add their extra layer of protection.
Sure. Well, those all sound like great ways to make sure that this is going to be helpful and not harmful. Tell us a little bit more about your upcoming clinical trials, because I know you guys are a little earlier than our other 2 presenters. So I'd love to hear more about those.
Yeah, so we're a little bit a ways away from the clinical trials. So we're still going to have to go through a lot of preclinical development before we get there, and we are expecting to get to clinical trials, or to start preparing at least the clinical trials in 2026. The area of engineered live biotherapeutic products or engineered bacteria is a fairly new one. So the regulatory strategies to be able to get into the clinic is something that is still sort of being developed by most of the regulators, both in the US and the UK. But we are in conversation with them already here in the UK, so that we understand exactly the points that we need to follow to get into the clinic in terms of the analytics that we need to have in place for the production of the bacteria, and all of the efficacy and persistence studies that we need to do in mice in order to get there. And particularly the fact that we are able to get to the clinic by using both mouse models of cancer, but also patient-derived material. That's really important, that we're collaborating with this group here in London that has developed models where you can study extracellular matrix integrity in material derived from cancer patients. So we can both have efficacy data and biodistribution safety data there. And through that, and through the work that we're doing in collaboration with the Pharmabiotics Research Institute, which is a specialist regulator consultant in this area, we hope to be able to get to the clinic within the next sort of 5 years.
Fantastic. No, that's really exciting. So we have a question in Q&A, and it actually is a take on one of the questions I was going to ask around. I'm assuming this type of platform could be used to treat other diseases. And Dr. Silverman asks, kind of in another direction, what do you have any news about insulin producing genetically modified bacteria to help treat diabetes?
And that's a really interesting idea. I'm not aware of anyone that is working on that. I know that there's a few companies that are working on bacteria for different diseases, like infection diseases, for example. We're very much focused on cancer, and that's because the properties of the bacteria that we are working with, what makes them really, really attractive to us is their ability to colonize these tumors and to be attracted to the environment that you find in these tumors. So we're using them as a means to an end rather than the actual objective of the company themselves, because we know that through them we have the capability of targeting the extracellular matrix directly in these tumors. Now that doesn't mean that they can't be used for other things. I'm sure they can, but the localization of them is really, really important for our strategy, and that's why we're targeting cancer specifically. And we also do have the plans in the future to additionally engineer these bacteria to not only target the matrix, but also deliver the immunotherapy drugs, so that we don't have to use these bacteria in combinations with anything else, but we can use them as single agents.
That's fantastic.
Excited about.
Yeah, very exciting. Well, Pedro, thank you so much for being here tonight and telling us about Neob Therapeutics. I'm going to invite everybody here in just a second to join us in networking, because what I know about these presentations tonight is it just brings up more interesting and exciting questions for me. As I mentioned earlier, don't forget to fill out the poll everywhere poll, because that feedback is really important for our CEOs and our founders. And tonight, again, I'm just going to give a couple of announcements, but please join our speakers and our networking lounge. To join one of the tables, you're just going to look for their logo on the table, click on a seat, and you'll enter the breakout room. We will have follow-up communication and email that goes out with all of the contact information and links to the videos from this evening. We also have a Pitch Club LinkedIn group that you can join us at. And we will load the program not only on the AngelMD website, but also our YouTube channel. And I think we had a link to that in the chat as well. As a final reminder, this will be our last Pitch Club event of the quarter. We're going to take the summer off. We're going to return in September for some fun new additions. And as Mike mentioned, we have information about the exciting new syndicate program and AngelMD Academy. Our first class is on Thursday. We'll put that information in the follow-up email as well. Thank you so much for all being here tonight, and we look forward to having you join us in our networking lounge.